Turn off the lights, please.
Hi, Mommy.
So, I'm posting it just like that. I'm not making any changes. So when we're taking a look at those chromosomes and how they move around, and the whole event of creating a zygote of sexual reproduction, there are consequences to that. And as we started to discuss earlier, Mendel discovered those consequences. Once he started doing those crosses, he noticed that there were, he called it agents, right? Inheritable factors. He called them factors. I don't know what exact word he used, but he didn't know the word gene. He didn't know the word allele. And there's several things that he learned from his statistical analysis. And understand, all he did was collect data and from growing peas and recorded it, and then said, well, what does this mean? And did work backwards from his data. And he said there's these factors. There's one that's going to be dominant. They seem one tends to be recessive, and they seem to be inherited independently. Or what he called independent law of independent assortment. And you know why that is today, because during meiosis, as you saw in two different activities when we did meiosis, and even some of you saw on your version of the test, when you're looking at a meiotic event and you have two homologous chromosomes in two different cells, right? So maybe this is mom's and some of dad's here, and this one's dad's and some of mom's over here. What you get is which chromosome went where. In other words, when you have these chromosomes that you had chromosome 1, and then you had chromosome 2. It's a smaller chromosome that also had moms. Maybe this one got dads on this side and moms. And the other one got moms with some of dads. So when you have two chromosomes, there's no way of knowing which chromosome went to which cell, because they assorted. Chromosome 1 from mom went here with a little bit of dad's on it because it crossed over, and chromosome 2 from dad went in this cell. So there's no way of knowing in meiosis in which direction they went. So that increases what? Genetic diversity. It's an important aspect of sexual reproduction. But when you end up with the fourth cell, or the second division in 4 cells, then you get here chromosome number 1, it's all moms. Chromosome number 1 with some of dads and the rest moms. Chromosome number 1 here, that's all dads, and chromosome number 1 over here, that's some of moms and mostly dads. And then you have chromosome number 2 over here, that's all dads. Which one did it go into? Maybe it went all the way over here. Maybe this chromosome number 2 went over here. And then the other chromosome number 2 that had a little bit of moms and some dads went over here. How do you know which cell got it? Which? You don't. It's completely random. So you just don't know what the combination of cells you're going to get. Maybe this one got all moms, and this one here got the one that had a little bit of dads and the rest moms. And so you get all kinds of mixtures that can happen, which is why it's difficult to predict what is going to end up happening in the end as far as what kind of person you're gonna get, or what kind of plant you're gonna get, or what kind of dog you're gonna get once you have sexual reproduction, because the whole point of sex is to mix up that DNA. So you just don't know what you're gonna get. So, again, looking at this dominant recessive inheritance, that's all Mendel looked at. He said it's either dominant or recessive. And all of you, I think all of you have said to me that you've covered Punnett Squares in middle school. Is there anyone in here that hasn't? Just a double check. So you did not. Only 2 people. You went over it when? Oh, you did it in Mississippi, but not in 8th grade. Okay. So most people have. So there's about 2 or 3 of you that have it. But when you look at it, what are you doing when you're doing this Punnett Square? Who can tell me, when you're looking at this Punnett Square, what is it that you're really looking at? Yeah.
The probability of getting one type of gene.
One type of genome versus another. Okay, that's good. That's a good answer. Yeah. 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 So you're both answering very good answers. So how, in sexual reproduction, where are you going to get these genes? From where? From parents, correct. From what specific kind of cell from parents? Sex cells. So there's 2 kinds, and let's just go with humans, but because there are all kinds of options when it gets to plants and fungi, and so let's not even go there. Let's just talk about humans or mammals. Let's say mammals. What kinds of sex cells do you have?
Sperm and egg.
Sperm and egg. So really what we're talking about is a sperm or an egg, right? So when you put letters, keep that in mind, right? Keep that in mind. Because when we're looking at sperm and egg, remember, what are we really talking about? I'm just trying to get this in perspective for you. You're talking about a DNA molecule here, and there's a gene on there. There's a gene. You know, this is one gene, and then there's another gene here, and there's another gene here, right? So there's all kinds of genes on this DNA molecule. That's the chromosome, right? It's long. There's one copy there. It might have a mixture of mom's and dad's DNA. Their father's mom's and dad's DNA, right? And the same thing's going to go over here with the mom's eggs. And then, so what mixture do you get? Which did you get? Did you get, let's say dad is homozygous dominant. Homo means what?
Same.
What does zygous mean? We're making a zygote. We're making a zygote. Zygote, right? Which is a fertilized egg. These are 4 possible zygotes. Homozygous means they're the same. You have 2 of the same. So dad is homozygous. So he was a zygote that had both the same genes, both the same versions of the genes. He was that zygote. So for this gene, so he was homo, same, zygote for this gene. Is that clear? Homo means same. Zygote means fertilized egg. So the father was homozygous dominant. That's right, homozygous dominant. So what would that be? Let's use the letter A. A for, I don't know, what do you want to say? What trait? Eye color. Let's say brown. All right. This is not the case. Eye color is actually very confusing. Color in general for any beast tends, for humans, any kind of color, skin color, eye color, hair color, all is very confusing because there's many different genes involved in it, and there's very different, gives you a lot of shades. That's why you don't, whenever you have dominant recessive, well, how many different kinds of traits do you have? Well, you have one trait, but how many different versions of that trait do you have? You have dominant and you have recessive. 2. You have the dominant or you have the recessive, right? So how many eye colors do you have? 2. You've only seen 2 different eye colors? One eye color? In the human population, how many different eye colors are there? I mean, forget about contact lenses, more than 5. I mean, there's green, there's different hazel, there's brown, there's real dark brown, there's light brown, there's blue. There's multiples. So that tells you that eye color, by definition, that tells you right away, eye color is not dominant and recessive. Right? Because dominant and recessive is only one or the other. That's it. So skin color is the same thing, right? There's not one or the other. There's many different shades of skin color. So there's no dominant or recessive. However, interestingly, there is a recessive form of eye color and there is a recessive form of skin color. That recessive form is albinoism. Albinoism is recessive because either you're an albino or you're not. Right? Either you're albino or not. So we can change this from brown eye color, and we can use any color. We can put A, instead of putting, we can say not albino. It turns out that the albino is a master gene that controls all pigment in an individual, and almost every organism has an albino version. There's a master gene that controls all the different genes that if it doesn't work, no pigment is made. So a big A anything, right? Big A, or what? What's another option? Big A, little a, they both are what? Both lead you to not albino. But if you have a little a, what do you have? An albino. An albino individual is someone who has no pigment. So their eyes are red, their skin, hair, they have no color. All right, so very pale. And that goes for polar bears, by the way, are not albino. They have pigment in their eyes. They can have, there is an albino polar bear. They don't, but it's hard to tell, obviously, because polar bears are white. All right, so if dad is homozygous dominant, what is he? Big A. What's the second? What other version? What are these? These are the possible genes he could. Big A and Big A, because that's all he has. So homozygous dominant person only has those to give. That's it. There's nothing else to discuss. Now, what if mom was an albino? So mom would have little a what? Little a, 2 little a's. That's all she could give. So I know right away in this gene combination, homozygous dominant with homozygous recessive. By the way, this is what? Homozygous what? Recessive. Right? So now mom is mom, which we kind of give the symbol this way for mom. And dad, let's just use red for mom. And dad has, they usually do this for dad, right? So those are 2 kind of standard males, female symbols. So homozygous recessive for mom, why is she homozygous recessive? Because she only has 2 little a's to give. As a zygote, she was homozygous for this gene, but she was recessive. Does everybody see that terminology? All right. So homozygous dominant for dad gives you 2 big A's. Homozygous recessive for mom gives you 2 little a's. So I know from this gene combination, if you look at it, you can go ahead and do the Punnett Square if you want. What are the 100%? 100%. This is easy. 100% of all their children's genotype is big A, little a. Big A, little a. So all of these are big A, little a, big A, little a, big A, little a, and big A, little a. But because of that genotype, the types of genes, what's the phenotype? Which is what you see. 100% as well. 100% what? Not albino. So a lot of you said non-albino for the first question, and you're right. This is 100% non-albino, but that's the phenotype. Make sure you write that down in your notes and you understand that, expect that as that topic as one of the quiz questions tomorrow. 100% genotype is big A, little a, across the board. 100% phenotype is not albino. So what do you see if you have this genotype? You see not albino. Understood? All right, so let's look at all the different possibilities. So that's if dad was big A, little a, big A, and mom was little a. So 2 homozygous, 1 dominant, 1 recessive. Another word for someone who's homozygous, dominant, or recessive, they'll use this term, you'll see it on tests, is true breeding. True breeding. So that means, remember, these studies were first done with plants and animals. And what do we do with plants and animals? We breed them, right? In fact, we call different dogs different dog breeds, right? So we breed dogs and animals. Obviously, we don't breed humans, but we call someone who's homozygous dominant true breeding, someone who's homozygous recessive true breeding. And we'll go into what Mendel did to come up with that. It's called a test cross, and you'll see what I mean by that. Okay, so if we have here a Punnett Square, and we look at what would happen if you had dad was not homozygous. What if dad was heterozygous? And so what, by the way, here's another way I could ask a question here. What's the chance, and I'm asking this question out loud, and you know you have a quiz tomorrow, right? So you should be making that connection for yourself. So what's the chance that any of their children, this first group of albino, non-albino, getting together, marrying, having babies, what's the chance that these individuals could have a baby that's albino? 0% chance. So it's 100% not albino, 100% big A, little a, but that also means there's a 0% chance of having albino child, correct? All right, I hope you get it. So what if dad's heterozygous and mom's an albino?
I mean, you got 2 different ones.
So dad is what?
A big A, little A.
Instead of big A, it's big A, little a, and mom is little a, little a. Go ahead and do that Punnett Square and tell me what to fill in, and then let's discuss the percentages. Right, so let's go ahead. What answer do you have?
50%.
Well, give me the fill in the boxes first.
A,
Big or small?
big A, little a, little a, little a, big A, little a, little a, little a.
All right. So doing the Punnett Square, that's done correctly. You say 50% what?
50% is albino.
Well, let's start with our genotype. Let's talk about genotype.
50% are recessive, 50% are...
No, genotype. I need someone to give me the full term based on what I showed you, what I said, use the words I used in the notes a minute ago. Go ahead.
50% are homozygous and 50% are...
Homozygous what?
Homozygous... Dominant.
Dominant.
I think you're homozygous recessive.
And 50% obvious. I think it's obvious, but go ahead and write it down if you want. If 50% are homozygous dominant, then the other 50% are probably homozygous recessive. Because it's 50%. So, but, okay. So that's an answer. That's genotype. That's correct. Now, what about phenotype? All right, question first.
How would that be homozygous if it's big A and a little A?
Oh, yeah, that's good. That was a good one. Homozygous recessive is true, but that's not homozygous. I'm glad you caught that. It's right, it's heterozygous. And there's no dominant or recessive. Heterozygous has both. I'm glad you caught that.
Oh, you're welcome.
I was confused with
I'm just not that way.
I don't know. I need more coffee or something. All right, go ahead. Heterozygous.
the phenotypes are half are albino. So 50% albino,
50% albino,
50% non-albino.
50% non-albino. Everybody okay with that? Again, the phenotype is what you see. The genotype is what the gene combinations are. What are the allele combinations? Remember, alleles are the versions of the gene. Are we good with all that? Phenotype is what you see, the trait. Tall or short, black or white, orange or yellow, flower, 3 petals in the flower or 4 petals in the flower, I don't know, fruit or no fruit, whatever it is that you're looking at.
Did you know
The consequence of having that genotype is some kind of phenotype, usually, right? All right, we're good so far? Any questions? Which one? You want to see phenotype here? You want that?
Yeah.
50% albino, 50% non-albino. Are we good?
Yes.
Yeah. Heterozygous. That means you have one dominant and one recessive, right? Are we good?
Yes.
All right, so let's go on. So so that's an interesting combination. Now, what about 2 people? What if one of these kids here marries one of these kids here? Right? One of these kids here marries one of these kids here. So let's do it. They're both what? What's this kid here? What is this? Describe this kid for me.
Heterozygous.
Heterozygous. Is it not albino, heterozygous? And this person here? Same, heterozygous, not albino. So if they mate at big A, little a, cross with big A, little a, cross meaning they could have babies. What are their combinations? Tell me. Yeah.
How would you choose between the recessive one and the big A little A? Because there's a 50% chance for both.
I don't know. They fell in love. I don't know why did 2 people get
No. I'm saying, why did you choose big A little A instead of the little A little A
for this cross?
for the second one? Because there's a 50% chance for both. I don't understand why you just chose.
Okay, I don't
Oh, you
understand.
just chose one?
Yeah, you just chose it, like, as an
Oh.
example.
Why did I choose this one and why did I choose this one?
No.
She was saying, like, why you choose the big A, little A instead of the little A. If it's a 50% chance, it could be both. But you're just using it as an example.
You mean for this cross?
Yeah.
Is that what you're talking about, this? You're saying, why did I choose this one for this cross?
Yeah, instead of the little A little
Right, I have my reasons. You'll see in a minute.
I know what you're doing.
Yeah. All right. Are we good?
Yeah.
All right, so everybody do it?
Big A,
All right. Big A, big A,
Big A, little A.
big A, little a.
Big A, little A. And then little A, little A.
Now this combination, which goes to the question that was asked a minute ago, is why did I choose big A, little a, 2 heterozygotes? Because when you cross 2 heterozygotes, you get kind of a different combination. The genotype, you'll notice that in both these cases, you have 100%, the genotype matches the phenotype. Here the genotype matches the phenotype, correct? 50% heterozygous and 50% homozygous, you get 50% albino, 50% non-albino. But here they're not going to match. So you have genotype, that's right, you got it. Genotype What's the genotype here? What percentages? Yeah.
25%.
Anybody? Yeah. Speak. 70? No. No.
25%.
50% what? What's 50% what? Heterozygous.
And then 50%.
25% homozygous dominant, and then 25% homozygous.
25% homozygous. Homozygous what?
Dominant.
Dominant.
25% homozygous recessive.
And 25% homozygous. Recessive. So you have, why 25%? Each of these squares equals 25%. 25% heterozygous plus 25% heterozygous, 25 plus 25 is 50. Are we all good with that? Here's the dominant, homozygous dominant, two big A's. That's only one square, that's 25%. And this is homozygous recessive, this only one square is 25%. So you have three different percentages when it comes to genotype. But interestingly, the phenotype, you don't have that many options. How many different phenotypes do you have? It's dominant recessive, so you only have two possible phenotypes.
50% albino.
That's right.
Yeah, 50.
Well, it's not 50-50, no. Look at it. Tell me. Yeah.
It's 75% non-albino.
That's right. 75% non-albino
And 25%
and 25% not albino. Why? Because, guys, look at it. If there's a big A, it's albino. I mean, it's not albino. So there's a big A here. So this one's not albino. Here's two big A's. They're equally non-albino. And here's a big A, little again, so that's non-albino. There's only one that's albino, and that's this one here. So only 25% are albino. So notice that these percentages are determined by the statistical probability that you could possibly get. There's only certain possibilities given what you have. So if you change what you have, you change what the possible outcomes are. You cannot, this is going to come into play when we do our pedigrees. Has anybody done pedigrees in middle school?
No.
Okay, so the answer is no then. Okay, so when we do pedigrees, this logic is absolutely mandated. If you can't figure out this logic, that you can only give what you have, then you won't be able to, you won't understand pedigrees.
Wait, say that last part again?
Go ahead.
Like I understand, like can it so say like a different, not this, but like a whole different child or something? Can't they have like if like instead of 75 out of line, can it be like 25 on line, 75 out of line?
I'm not understanding what you mean.
No, I'm saying like, alright, say like this child with others, and maybe somebody else, be like a child.
So yeah, so it all depends on which kid. If one of these two kids marries someone else who's heterozygous, so that's two heterozygotes, you're going to get this possibility. So you're going to get 25% possible albino, 75% non-albino. Those are the odds. You understand, and this is really important because guys, someday some of you are going to get married. You're going to want to have kids. Somebody in here might have sickle cell trait. You might marry someone with sickle cell trait. Some of you in here might know, because you do a DNA test, you might know you have a gene that increases the risk of breast cancer or heart disease or any number of diseases, right? So if you marry someone else, you're going to get married, you're going to want to have kids. Some people go, if they know that they have this family disposition for having these problems, right? For instance, we know that Angela Jolie, with the breast cancer gene. We know that there are people that have schizophrenia that runs in the family. If you know that runs in the family, you're worried that you don't have it, you're good. But you're worried that your kid might have it because you might have a what? A recessive gene. So you're going to go to someone called a genetic counselor. So it's one of the possibilities you can do. There are people across the street, actually, there's a whole clinic where they go and they get a blood work, they get tested, and they go for genetic counseling. Yes? It can be inherited. Not always. Not all schizophrenia. There is a it's what we call how likely are you? Understand, in many, many diseases, you get what's called the predisposition. So what that means is that you're at increased risk to get it. Not that you're guaranteed to have it. So Angela Jolie had this gene that made her at increased risk to having breast cancer. She chose to take care of that quickly. But she wasn't guaranteed to have breast cancer. She just had a higher percent chance of getting it. Yeah.
So it's like things like heart murmurs, is that like passed down to you?
Heart murmurs can be passed down. Not all. Now, when it's be very careful. Everything we're talking about with genetics, sometimes you can catch, sometimes it's not catch. Sometimes that you do inherit the predisposition. Sometimes you're just unlucky.
I know, like, my mom got one and I got one, my sister didn't get one.
Right. So I don't know if it's inherited. I don't know. It depends on why it occurred. Some people are born, for instance, with cerebral palsy. My aunt was born with cerebral palsy. She was a twin, and she had cerebral palsy. Occurs when there's lack of oxygen to your brain as you're being born, and you get brain damage. The brain damage might be just motor, or it could be it could involve your thinking. It could involve your ability to learn, or your ability to see. You could be blind, or it could be a lot of things. Anything that your brain controls, if you have brain damage, it could impact that. So cerebral palsy is not inherited. Right? You don't inherit cerebral palsy. So just because you have a mental illness or a mental disability does not mean you inherited that mental disability. It could be maybe somebody's mother smoked while they were pregnant. Maybe somebody's mother was exposed to a chemical. They were working in a factory and were exposed to a chemical. Maybe somebody's mother took a drug that caused a birth defect. These things occur without there being a genetic link. Yes. Go ahead. Yeah.
So like this piece on Punnett Square is like impossible outcomes. It's like totally different.
That's a good question. So Punnett Squares are talking about possible outcomes. So let's assume that you are heterozygote for that albinoism gene. I don't know who is or who isn't, but you marry somebody who is also heterozygote for that gene. You have 4 babies. How many of them will be non-albino? If you are heterozygous, so you marry someone who's heterozygous, and you have 4 babies, how many of those babies will be non-albino?
3.
You say 3. What do you say?
It's not determined because it's just like a probability.
Excellent. You understand well. So these are just probabilities. It is not determined. This is genetics. It's not determined that you're going to get that, because who knows which sperm your husband gives? Who knows what egg you make? You actually provide. It's all a matter of luck.
So it could be like that 125% chance that child is albino.
You could have 100 babies. You'd be tired by the end, but you have 100 babies, and with the same man, and have 0, and none of them are albinos. They're all non-albinos. You could have 100 babies with the same man, and they can all be albinos. You just can't tell. Now, how likely is it? Very unlikely, right? These are percentages. Yeah.
So let's say, like, you get it when, like, after you have kids, is that typically,
No, wait.
Because, okay, look, so after I was born, like, my dad got it, but he got, like,
Got what?
schizophrenia. But he got drugs, but he...
No. Don't misunderstand what I say. I say there's a genetic link to some forms of schizophrenia. Schizophrenia is a big, you understand what I'm saying? It's a big mental illness. It encompasses a lot of different factors. Again, it could be environmental, right? You could have the gene for a particular disease. Like, let's say my father, for instance, had a heart attack when he was 40. So I could have the gene that would predispose me to having a heart attack. What's the difference between my father and I? My father smoked and I didn't. So now you have the environment impacting the expression of that gene. Do you understand what I'm saying? So there's two questions. Did your father give you the gene? And the second question is, if it is, well, first, is it genetic or was it environmental? So now we're not going to discuss your personal issue. I'm just telling you the questions you want to ask, right? So this is what genetic counseling is all about, by the way. Do you hear me? This is why you go to a professional. Is that clear? You're concerned, and you're right to be concerned. I'm concerned about heart disease, and I'm right to be concerned, right? Because I was obese and my father had one, so I should be concerned. So what you need to do is you need to go to a genetic counselor, and that goes for everyone, for all diseases, right? If it's genetic. So the first question is it genetic? Secondly, what form of the disease did my ancestor have? It could be a grandfather, it could be a great uncle, right? What form of the disease did they have? Is it genetic? If it is genetic, do I have the gene? Is the gene even known? Some genes we don't know. We just look at the pedigrees, the family tree. By the way, a pedigree is a family tree. We look at the family tree and we say, wow, your great uncle had it, your great great uncle had it, your great great uncle had it. It's probably genetic. We don't know what gene it is, but it's probably genetic. There's a high likelihood and they do a statistical analysis to figure that out. So this particular form of the disease, again, what did I just say? Particular form of the disease. Some forms of mental illness are caused by the environment, i.e., for a good example of this is doing drugs. Doing psychedelic drugs can give you a mental illness, right? That has nothing to do with genetics. So you have to figure all that out. And then you have to ask yourself, is it dominant? Is it recessive? Is it multi-gene inheritance? Are there many factors that come into play? That's why you go to an expert to ask those questions. All right? Now, so when we're talking about dominant recessive, life is good, and you see it's pretty easy. Do you all agree?
Yes.
So tomorrow's quiz, if I give you one of these, it should be pretty easy, except, and this is where the exception comes in, is there anything easy in the world?
No.
No. There's nothing easy in this life. I don't care what kind of easy button you have. Do you remember that commercial, the easy button?
No.
That red easy button. I used to have one. I put it on my desk every time my kid said, "Why?" I hit the button and say, easy, baby.
Mom ain't say that.
No, he just made that up.
No, I would say, easy, baby. And the thing would go, Anyways, whatever. I know. What about when you have two genes? What about instead of albino, not albino, what if you're worried about albino, which we just talked about as being dominant, either A or little a, right? If I put A blank, you understand that means A, big A, or big A, little a is going to give you a non-albino. And little a is going to give you albino. Does everybody understand that notation? Okay. Are you wearing a hoodie? Are you wearing a jacket? So, what if someone's albino, but we take into consideration, what if we want to ask, not just albino, but how about albino and tall? So I'm going to say there's two, just for argument's sake, and let's not use tall, because it turns out that, again, well, let's talk about a plant being this way. Humans are not, are tall and, there's not tall or short, right, with humans? Do you agree? You got all kinds of heights. So what do you know about height with humans?
Maybe 60.
It's very
There's a variety, so you know it's not dominant recessive, right? You know it's not dominant recessive if you have many options. So it's not tall, but it's not an animal. Let's say it's a plant,
That's gonna be all right.
a flower. Let's look at a flower. You're right, it can't be an albino. It can't be an albino, so let's call it, because if there's albino plants don't survive, why?
Because they don't got color.
They don't have color, so they can't have green, they can't do photosynthesis, they die. I mean, that's it, that's easy. But let's call it red. It's either red or not. And let's call red being, big A is red, and little a is white. And with tall, it's either gonna be, the plant's either gonna be big T blank, and it's gonna be tall, or it's gonna be little t, and it's gonna be short. All right, so you have two genes, two traits. Uh-oh, now we got trouble. You don't think we have trouble?
Then we gotta make like a bigger Punnett Square, don't we?
That's right, we gotta make a bigger Punnett Square.
It's the same thing.
Is it the same thing?
It's
Is
not
it?
the same thing, but it's.
So you got two, what are the different combinations of AT? So you have A, big A, or you can have a big T, or you can have what?
Little way.
Little a, big T, or you can have what? Big A, little t, or you can have what? Little a, little t. Oops. Right? So it can get complicated, right? Do you see why it's not easy? Yeah. Yes, of course. Do you agree? Or would you?
Yes, yes.
Okay. So we're going to go ahead and do a Punnett Square. It's called a dihybrid cross. Pay attention because I only have time to do it once. If dad is heterozygous for both, heterozygous. So dad is big A, little a, big T, little t. And this plant is crossed with another heterozygous, big A, big T, little t. What are the possible combinations that you can have? So here's what I'm going to tell you to do. I'm going to tell you to do FOIL. And I know you covered it in Miss Simpson's class. So let's see if you remember. I'm going to tell her if you forgot. First stop, when I'm done here, before I can do my walk, fourth period, I'm going to stop at Miss Simpson and let her know.
She is not neither.
I got a question.
I'm using W.
What are you doing?
I use Punnett Square in her class.
Oh, good. Yeah.
When we do FOIL with this, do you want us to like square it or to?
No, I want you to do FOIL. Somebody tell me how to do FOIL. Help me.
We do W.
FOIL means what? FOIL. F-O-I-L. First, which is what? Big A. Big A what? First, big A, big T. Right? Big A, big T. Then what's the second? What's O? First, outer, big A what? Little t.
I'm saying.
I don't care how you do it. Hey, listen to me. Listen carefully. I'm showing you what I feel is the easiest way. I don't care how you decide to do it. As long as what?
It's right.
As long as it's right. As long as it's right all the time. Sometimes you can get lucky and get it right in the question you're given, but tomorrow you get another question, you get it wrong. This way you always have it right. This gamete, this individual, this plant can produce this gamete, or it can produce another gamete. What's the other gamete it could produce is big A, little t, right? Those are the two so far. So that's first and outer. Then what?
Again, little a, big
Then what?
Inner.
Inner, which is the little a, big T, right? So the third is big A, little t. Oh wait, I'm sorry, I messed that up. Little a, big T, and then what's the fourth possibility? The L. L is what?
little a, little t.
Little a, little t, the last. Little a, little t. I really don't have a problem if you have your own way of doing stuff as long as this works. FOIL works all the time. So if you're going to do dihybrid cross, one parent has these four possibilities. What's the other parent? What's the other possibilities for the other parent? It's the same thing. Because the other parent's also heterozygote. So those are the two possible gametes that these parents can produce. So how I have to now do a Punnett Square with those. So I would do big A, big T. This person, this gamete, this sperm is going to be big A, big T, or pollen, or whatever it is. And the other one's going to be big A, little t. I'm going down here, right? And then the other possibility is little a, big T. And the last possibility is little a, little t. So there's the four possible pollens, right? Four possible gametes that individual can give. Now I have to do the other parent. The other parent does what? Has a big A, big T, same combo, right? Big A, little t, little a, big T, and little a, little t. Of course, I have to go across now. Can you see how this is going to get complicated?
Yep.
It's really not that bad, but instead of just 4 squares, you have what?
18,000.
No, 16. Come on, 4 by 4 is 16. 4 times 4 is 16. You can handle that. Not 18,000.
Hi.
All right. So in the big A, these two, if this male gives this gamete and these two gametes combined, what do you get?
Capital AA.
The zygote would be big A, big T, big T. So this thing would be red and tall, correct?
Mm-hmm.
Then if these two get together, this one and this one, what do you get?
Big A, big T, little T.
Big T, little t. And the next one, big A, big T, and then big A, big T, little t.
Very time consuming.
It's gonna be, yeah. And the next one, big A, but there's some shortcuts I'm gonna show you.
Big T.
I just want to see. I want to show you where you get these from. Big A, big T, little t. Big A, little t, little t. Big A, little t. Oh, sorry, big T. Big T, little t. Big A, little t, little t. And then of course, big A, big T, big T. Big A, big T, little t. Big A, oh, sorry. Little a, big T. Sounds like a rap, doesn't it? Big
Hi,
A,
Riley.
What's your name? Betty?
little a, yeah, fatty, yeah, man. Little a, big T, little t. Big A, big T, little t. Big A, little t, little t. Little a, big T, little t.
All right, you're not wrong.
Little a, little t, little t. In genetics language, that's a rap. I don't know what that is. I don't know what you're saying, but I'm gonna go ahead. I'm just gonna go ahead and continue this.
Hit your pose real quick. Hit your pose.
All right, so,
Hey.
all right, so, hey, my generation came up with that, all right?
Oh my god.
It was the 80s.
Don't nobody use that no more. But
It was the 80s when rap started. Breakdancing in 80s. Just so you know, breakdancing and rap my generation started it. Done. Whatever. We used to scratch records too. When I say we, not me. My brother did. My brother did the rapping and the
Oh, that's why you want to
breakdancing. But
I hope he was a rapper like you.
not me. I have no rhythm, so I have no.
that's you. Hispanics have.
I have no
really
rhythm. I'm the only Puerto Rican in the world without rhythm. It's really
That's what I'm
embarrassing,
saying.
but it's sad. Yeah.
I like the way you dance. Do your hips lie?
All right, can we focus? All right, so let's do genotype, and now we get into the. I'm sorry. Now we gotta get into the exciting stuff.
My hips don't lie.
So
It's time to go.
is it time to go? All right. Dang, you're missing the fun stuff. All right, so you be ready to do stuff like this tomorrow on the quiz. Bye.
Bye.
So let's take a look at this. We get to have some fun with this. Now we have all this data. How many of these, what are the genotypic ratios? Genotypic ratios. Uh-oh. What? Let's talk about genotypic ratios.
It's the heterozygous and homozygous.
Okay, so the genotypic ratios for big A, big A. How many are big A, big A? Out of how many? How many do we have? How many possible ones? 16. So how many are big A, big A, big A, big T, big T? Big A, big A, big T, big T. How many are we? Let's see. Big A, big T, big T. I see one here. There's only one. So it's 1, 2, let's go to the next one. Big A, big T, little t. Big A, big A, little t. Or big T, big little t. Big A, big T, little t. There's 2. 2.
Oh.
So it's 1 to 2. Hopefully you start seeing a pattern. How many do you see that are big A, big T, big T? Big A, little a, big T, big T. 1, 2. So I see 2.
You put them together side by side.
Oh, it's going like a multiplication thing?
How many? Exactly. Very good. How many are big A, little t, little t? 1. So we got that one. We got these. Let me do different colors, might help. We got these.
No.
We got these. And we got
Let me make that green.
and we got this, right? Do we all agree that those are done?
Yeah.
There's 1 of these. There's 2 of these. There's 1, 2 of these. There's 1 of these. Is that correct? All right. So let's keep going here. Ooh, what's going on? I don't know.
And it's gonna be 40.
4 of them.
You think?
Yeah.
Okay. So we have 1 to 2 to 2. Let's keep going. So the next is big A, big T, little t. Big A, big T, little t. 1, 2, 3, 4. 1 to 2 to 2 to 1 to 4. So that's all. Oh, that's all of these and this and this. These are all right. Now, how many of these are now going to be big A, little t, little t? 2. 4 to 2. I'm running out of colors. That's this one and this one. How many of these do you see that are little a, big T, big T? One. I'm just going to go ahead and reuse a color because I don't want to do it again. How many of these are little a, big T, little t?
Two.
The two. And then how many of these are little a, little t, little t? One. Wow, that's a lot. Is that a useful? This is kind of it's called genotypic ratio. Is that useful?
Yes.
I mean, I guess it could. They're really useful for the regular monohybrid cross, the ones we did earlier in today. This dihybrid cross, I mean, yeah, you can figure it out if you memorized it, I guess. But the phenotypic ratio, now that's interesting. Let's look at the phenotypic ratio. How many of these are red? Red. Now listen. How many of these are red and tall? Let's see. This one's red and tall here. This one's red and tall. This one's red and tall. So that's 3. This one's red and tall. 4. This one's red and tall. That's 5. This one's red and tall. 6, 7, 8, 9. That's it. Correct? So 9 are red and tall. How many of these are red and short? Red and short, 9 to 2. Right? That's all there is. There's only 2 that are red and short. Does anybody disagree?
What about the big A, big A, and little t, little t?
Where? Big A, where? Do you see it?
The one that's not crossed off.
I said they're red and short. Little t, short. Big A, little a is red. Correct? So only 2.
You said red or short? Yeah, second.
Oh, red and short. So red and short, red and short. So 3, 9 to 3. Sorry about that. 9 to 3. Correct? And then how many of these are white and tall? White and tall. 9 to 3 to what?
Little A and big T.
2 little A's and any big T.
4.
3. 1, 2, 3. 9 to 3 to 3. How many of these are red, are white and short? 1. 9 to 3 to 3 to 1. This ratio is very useful. That's something you can remember. 9, 3, 3, 1. You can remember 9, 3, 3, 1. It will save you so much time if you remember 9, 3, 3, 1. 9 to 3 to 3 to 1. What does that mean? That means that you will have, what that means is that you will have 9 that for the phenotype will have both dominant traits, right? In this case, red and tall. You'll have 3 that are going to be dominant for one and recessive for the other. Is that correct? So what does that mean? Red and short, right? You'll have another 3 that are going to be dominant reversed, right? So they'll be tall and white. And you'll only have one. For every 16, you'll only have one. Only one that will be recessive for both. White and short. In this case. Remember, they're going to change the traits on you. It won't always be white and short, red and tall. It'll be different for everything. It could be sickle cell and not sickle cell, because sickle cell is dominant recessive. It could be sickle cell and something else is dominant recessive, if it's in humans. Correct? It could be anything that's dominant recessive will have a 9-3 to 3-1 ratio. If you know this and understand what the ratio means, your life will be a lot quicker when you're doing multiple choice exams. All right, have a good one. Wrap up, put away, and get out of here.